Anatomy of the retina. The retina, as you all know, is the innermost layer of the eyeball. It is thin and delicate and is transparent. That means we see the blood vessels below and the choroid that lies underneath of it. It appears, therefore, purplish in color. On gross anatomy, there are three distinct regions in the retina. The first is the area of the optic disc, the second is the macular area, and the third is the peripheral retina. The retina extends from the optic disc to the end of it, which is the ora serrata. Its surface area is almost 270 square millimeters. Here you can see the optic disc where the blood vessels are branching from it, whether these be the retinal arterioles or the retinal venules, and the central area here is the macula. The optic disc is a pale pink structure that is well defined and rather oval to circular in diameter. Its diameter is 1.5 millimeter. All the retinal layers terminate at the periphery of the optic disc except for the nerve fiber layer, which passes through the lamina cribrosa, which is in the center of the optic disc. Now the second area is the macula lutea, and this is comparatively dark to the rest of the retina. It's about 5.5 millimeter in diameter, situated at the posterior pole of the eye, and it's always temporal to the optic disc. It's also called the yellow spot, as lutea means yellow, and may be called the area centralis, being the central area. Its primary function is photopic vision, that is, vision in the light conditions. The fovea centralis is the central depression in the center of the macula, and it measures 1.85 millimeter in diameter and 0.25 millimeter in thickness. It is the most sensitive part of the retina. Here is a magnification of the central area of the macula lutea, where the yellow pigment appears amidst the purplish to reddish background. And you can notice that this area is avascular. The foveola is 0.35 millimeter. It's the central floor of the fovea, where the umbo is the tiny depression at the center of the foveola. So we can differentiate the macula as 5.5 millimeter, with the central part of it, the fovea, is 1.85 millimeter. The foveola is the most central part of the fovea, which is 0.35 millimeter in diameter. And finally, the most sensitive tiny depression in the center of the foveola, called the umbo. The periphery of the retina can be divided into the mid-periphery, the far periphery, and the ora serrata. Some authors like to add the near periphery. Microscopically, the retina is formed of the following layers from inside to outside. From the vitreous, we find the internal limiting membrane, followed by the nerve fiber layer, and then the ganglion cell layer with the inner molecular or plexiform layer, then the inner nuclear layer with the outer molecular or plexiform layer, and then the outer nuclear layer, the external limiting membrane, and innermost are the rods and cones, the layer of rods and cones that are inserted into the retinal pigment epithelium. So the light passes through all the structures of the retina to be picked up by the innermost photoreceptors, and then this light is transformed into a signal that passes through the nerve fiber layer into the optic nerve. Here is the innermost layer, where you can see the nerve fiber layer and the internal limiting membrane. And here is the outermost layer, which is the layer of rods and cones, and finally the retinal pigment epithelium. Actually, the retinal pigment epithelium is the outermost layer of the retina, as I said. It's made up of a single layer of hexagonal cells that contain pigment. It is firmly adherent to the underlying Bruch's membrane of the choroid and loosely attached layer of the rod and cone cell. It has got the subretinal space, which is the potential space between the retinal pigment epithelium and the retina. And actually, clinically speaking, when we say retinal detachment, that means separation of the retinal pigment epithelium from the retina, and subretinal fluid may accumulate between the sensory retina and the retinal pigment epithelium. The retinal pigment epithelium cells are connected to each other by zonule occludents and zonule adherents. Here are the tight junctions, as you can see, at the lateral sides between the very active retinal pigment epithelial cells, and they have the inner layer communicating with the Bruch's membrane, and the outer layer has got apical microvilli that communicate with the photoreceptor cells. The functions of the retinal pigment epithelium are renewal of the photoreceptors with recycling of vitamin A precursors and metabolites. The layer provides mechanical support to the processes of the photoreceptors and manufactures the pigments absorbing light, and the retinal pigment epithelial cells have got a phagocytic activity as well. Now we come to the layer of rods and cones. The rods and cones transfer light energy into nerve impulse, and the rods in particular contain a photosensitive substance called rhodopsin, where the cones contain the other photosensitive substance called iodopsin. The cone cells are concerned with photopic vision, and rod cells are concerned with scotopic vision. The number of cones in the eye are about 6.5 million, while the rods are almost 20 times that much. That's 120 million in number. Now we come to a diagram outlining the structure of the rod and cone cells, where each is composed of an outer segment, which is the photoreceptive region, and an inner segment, which is the synaptic body. Now we come to the structure of the rod. A rod is 40 to 60 micron in length, with an outer segment and an inner segment, as we said. The outer segment is cylindrical. That's why the name rod, composed of numerous lamellar discs formed of plasma protein membrane. And the discs are 600 to 1000 in number in each rod. The thickness of each disc is 22 to 24 micron. A disc contains 90% of the visual purple, and the outer segment is attached to the inner segment via cilia. A single cilium connects each outer segment to each inner segment of every rod. The inner segment consists of morphologically two areas, the ellipsoid and the myoid. And here is the ellipsoid and myoid of the rod, probably more pronounced in rods than in cones. And this area is connected to the outer segment by cilia, as we said, and through these cilia pass fibrils of microtubules formed of actin filaments. And this area passes through the outer limiting membrane into the nuclear area of the rods and cones, forming the outer nuclear layer. Here, once more, a diagram comparing the rods to the cones, and as we can see, the main differences are: first, the outer segment is like a rod in rods and like a cone in cones. The cones are shorter. The myoid and ellipsoid parts of the inner segment are more pronounced in the rods than in cones. And uh, the outer rod fiber arises from the inner end of the rod and passes through the external limiting membrane, then further swells into an area that contains the densely stained nucleus. The nucleus terminates further into the inner rod fiber, and the inner rod fiber ends as a bulb called the rod spherule, contrary to the end of the cone, which is the cone pedicle, as we shall see in the next slide. A cone cell is shorter, 40 to 80 micron in length. At the periphery of the retina, it's 40 microns only, and at the fovea, it's the longest, where each cone is 80 micron. Outer segment is conical, shorter than the rod, as we said, containing the iodopsin pigment packed in lamellar disc, and the number of discs here in cones is 1000 to 1200 in each cone. The inner segment and cilium have a similar structure to the rods, and the inner segment is directly continuous with the area containing the darkly stained nucleus, and a stound cone inner fiber runs from the nucleus and has a lateral process at the end called the cone foot or cone pedicle, compared to the rounded area of the rod, which is called the rod spherule. Now, the external limiting membrane is a fenestrated membrane extending over the whole area of the retina from the ora serrata up to the edge of the optic disc. Through it pass the processes of the rods and cones, and this external limiting membrane is formed by the junction between the cell membrane of the photoreceptors and the molar cells. Here is the external limiting membrane through which the rod and cone cells pass by the connection between the inner segment and the nuclear layer. The outer nuclear layer, as we said, is made up of the nuclei of the rods and cones. The cone nucleus is slightly bigger than the rod nucleus, and these nuclei lie in a single layer next to the external limiting membrane, and the rod nuclei form the bulk of this layer, except at the cone-dominated foveal region, as we said. There are variations in thickness of this outer nuclear layer. Nasal to the disc is formed of 8 to 9 layers and is 45 micron in thickness. Temporal to the disc, it's thinner, formed of only 4 rows, 22 micron. At the foveal region is formed of 10 rows of nuclei, 50 micron in thickness. The rest of the retina, except the ora serrata, it's only 27 micron in thickness, where there are 1 row of cones and 4 rows of rod nuclei. The outer plexiform layer is made of synapses between the rod spherules and the cone pedicles with the dendrites of the bipolar cells. The bipolar cells are in the inner nuclear layer, and this inner nuclear layer is a very thin layer and is similar to the outer nuclear layer, consisting of the following cells: the bipolar, the horizontal, the amacrine, the molars, and as well as the central retinal vessels and other glial cells. And this inner nuclear layer is not existent in the area of the fovea. Here, this is the structure of the inner nuclear layer, where we can see the molar cells, the amacrine cells, the horizontal cells, the bipolar cells, anastomosing with the inner segment of the rods and cones. The bipolar cells, as you can see, are cells connecting the photoreceptors to the ganglion cells in the next layer. While the horizontal cells interact with the photoreceptor and the bipolar cells, they receive input from the photoreceptors and use that input to integrate signaling and make adjustments to signals that will be sent to the bipolar cells and therefore regulate the activity in the photoreceptor cells themselves. The amacrine cells act as an interneuron between the bipolar and the ganglion cells. They receive signals from the bipolar cells and they are involved in the regulation and integration of activity in bipolar and ganglion cells. The Muller's cells are the predominant macroglial cells in this part of the retina, responsible for functional and metabolic support of the surrounding neurons, and therefore they consume a very high level of glucose. The inner plexiform layer consists of synapses between the bipolar cells and the dendrites of the ganglion cells, and again are absent at the fovea. As there is no inner nuclear layer, there are no bipolar cells, so there is no inner plexiform layer. And then comes the ganglion cell layer, which consists of the cell body and nuclei of the ganglion cells. This is the second order neuron, second to the rods and cones. It's again absent at the fovea, and there are various types of ganglion cells. The types of ganglion cells are classified in four main categories. The first is the WXY ganglion cells. The second is P and M ganglion cells, and P are classified into P1 and P2. Third is off-center and on-center cells. And finally, the monosynaptic and polysynaptic cells. Here we can see the ganglion cells, second order neuron communicating with the rods and cones via the bipolar cells. Now the nerve fiber layer consists of the unmyelinated axons of ganglion cells which converge at the optic nerve head. Its thickness is half to 2 micron. Thickness decreases from the optic disc to the lateral side, and the fibers show a definite pattern of arrangement. Finally, the internal limiting membrane, which consists of the following elements: the collagen fibrils, the proteoglycans of the vitreous body, the basement membrane, the plasma membrane of the molar cells, and finally other glial cells. Now we finally come to the blood supply of the retina, and the blood supply of the retina comes from the choriocapillaris of the choroid and from the central retinal artery. That is a double blood supply. The outer four layers of the retina are supplied by the choriocapillaris, while the inner six layers are supplied by the central retinal artery. The fovea is only supplied by the choriocapillaris, where the macular region takes branches from the superior and inferior temporal branches of the central retinal artery. All these make three plexuses. One is the deep vascular plexus in the outer plexiform layer. The second is the intermediate vascular plexus in the inner plexiform layer. And finally, the superficial vascular plexus that is lying under the internal limiting membrane. Thank you very much.